Welcome to today's seminar. I'm really pleased and honored to introduce to you today Indresh Srivastava. Indresh is the Vice President of Process and Formulation Development at Novavax, and he's also President-elect of the International Society of Vaccines. First of all, my name is Manon. I'm the current president of ISV. I'm really honored to have that role for you, but I'd like to start with a brief introduction of Indresh. For those of you who don't know him, I have worked with Indresh for about 10 years while we were at Protein Sciences. Indresh really helped there with the development of FluBlock, and when the company was acquired by Sanofi, Indresh continued to work there for a little bit. And after that, he joined Novavax. But before Protein Sciences, Indresh already had a long history of vaccine development, and you would not be surprised to see Indresh in meetings, knowing everyone either from his past at Chiron or Novartis or whatever. He's also the author of, or actually, I should say, actually editor of one book that has been published and another one that is in preparation. And he has authored many papers on vaccine development. So it's really an honor to have Indresh here and and he will today be speaking about the development of a thermostable vaccine. I hope that many of you were able to deal with the time differences, etc., since the US has now gone in summertime. But without further ado, I'd like to open the floor to Indresh. Indresh.
Well, thank you. Thank you very much, Mano, for a nice introduction. Yes, I think I worked with Mano for 10 years, but I think I know Mano since 2003. And Mano, as usual, she is so highly organized that the card I gave it to her in 2003 meeting, she gave it back to me when I joined Protein Sciences. So that's our link. I thought that this is going to be a good topic to talk about it. I think we are doing manufacturing and everything else, but this is a very important aspect. So before going too much into it, I just want to go to the next slide. This is what I want to show that what I will be covering into. I think is just probably for this audience, it is not needed to remind that the thermostable vaccines have tremendous advantage, both in terms of the compliance and also stability and controlling the cost. So that's the first bullet. The second bullet, I think we are going to be talking about the approaches to develop thermostable vaccine. And the first bullet is about the structural biology and molecular biology approach that we have used to develop a stable flu vaccine and liquid formulation. This is the work that I did when I was with Mano at Protein Sciences, and I think it's a large team of Protein Sciences that contributed in the successful completion of the work. So I will run through and then share the information and the knowledge that we gained from that approach. Then the other topics are that I think we are using the liquid formulation. The field is using liquid formulation. The field is also using lyophilization. And then we will touch upon the needleless patch delivery because that's also developing a thermostable vaccine. At the end, I will just give you a little bit of my 2 cents where we are in the field. So going to the first topic, I don't need to see it. This is what is very important to say that what value the thermostable vaccines offer, because vaccines are the ones which are protecting millions of people. Different vaccines in pediatrics portfolio, travelers portfolio, other diseases portfolio, and these are the vaccines are the ones which are the medicine given to the healthy people. So obviously the bar is high, the cost is high, but the one of the major limitation is that most of the vaccines that are approved to date, they are either stored at 2 to 8 degrees or they stored frozen, and that requires the cold chain for distribution. And also probably the one of the major impact is that I think with the temperature excursions, these vaccines lose the potency. So they become kind of waste. Um, if we have an approved thermostable vaccine, then we can store it at the room temperature or maybe even higher temperature, and that will facilitate distribution and the cost as well. I just wanted to show you, I think in the next slide, a survey and was published. Going to the next slide, please. Me, yeah. So here, I think the survey was done in different countries. You will see that Brazil, China, India, Peru, Philippines, Tanzania, Zambia, and Zanzibar. And what you are seeing here is that a number of doses were destroyed because of temperature excursion. Either it's heat exposure or it's freezing exposure or both. So there is a huge amount of vaccines that were wasted, and that's a constraint. Going to the next slide, I think what we are targeting towards is that thermostable vaccine can remain stable and retain their potency even when expressed in the fluctuating temperatures. That's one part. By removing the strict requirement for the cold storage, thermostable vaccines can overcome the logistic barriers and expand access to the immunization programs in these underserved regions. And the other part is that I think it will also improve the compliance. People will come, more population can be covered, and it's also global health equity initiative will also can be supported by the thermostable vaccine. So I think it is the right time to start focusing. And I think a lot of academic institutions and also some of the industries, they are looking to develop thermostable vaccine. And in the second half of my talk, I will cover those things. Just to give you a perspective, these are the approved vaccines, and I think you will see that there are very few. There is no thermostable vaccine that has been approved yet, as per my knowledge, and I could be wrong. If I'm wrong, I will please let me know. But there are some vaccines that are partially thermostable. So post-thaw, post-storage, they have some stability at the room temperature. So again, I think it's going to be very important to capture this part and then develop a thermostable vaccine. Going as I promised, I think the second part, I think it's what approaches has been developed successfully and evaluated in either animal models or in the clinical trial. That's the second part I will cover more in detail. As I said, I think this is the work that the team started. I think protein sciences team, I think Kathy Holtz was the one who was leading that effort. And what we realized that our H3HA, they were losing potency very quickly. I think initial loss of potency was as high as 40%. And we spent a little bit of time in terms of understanding it, why there is so much loss we are getting it. And what we have found out in the various studies that we have done, that it is a cysteine at position 549 that was getting oxidized and causing the multimer formation. With the multimer formation, you change the conformation. The conformation is changed for the protein, so it will not diffuse that well in the SRID assay. And that's that's what I think we published that work in Journal of Pharmaceutical Sciences. This is with Rasmida's group at University of Kansas. So what we decided to do at that time, that okay, can we look at it, the number of cysteines that these proteins had, and that I will cover in the next slide. But we identified that there are 5 cysteines in the transmembrane and the cytoplasmic tail. The number varies with the subtype. Like for example, H1 had 3, and B had 2, and the H3 had 5 cysteines. So we said, okay, can we just make the changes So the team mutated or introduced the variant, changes 2 cysteines, 3 cysteines, and 5 cysteines. So we made three constructs along with the wild type one. And then we did the purification, expression purification, and then we did the characterization. We did quite in-depth characterization. That's what I think we will show you the data. We look at the purity, we look at the potency, we look at the hemagglutination activity, we look at the homogeneity, we look at differential scanning fluorometry, so we can also look at the TM, DLS. So we just did all those things to fully characterize the proteins and then put them on the stability. And that's where we had the surprise that I think by changing this, by changing the cysteines to another either serine or alanine, we're able to create a highly room temperature stable H3 vaccine. We have also done later on, I think we did, the team has done the same work for H1 and B, but we did not see the same. But if you combine them together, we were getting the thermal stability. So without any more delay, I think this is the slide. As you see, I think in the block squares, I think these are the cysteines. These are the 2 cysteines in the cytoplasmic tail. They are conserved across all subtypes, means H1, B, and H3. We look at the number of H3s and number of H1 and Bs. Here are the, for H1, as I mentioned, the three cysteines in the cytoplasmic tail, they are they are conserved. And in the B, there are the only 2 cysteines, and for H3, these are the 5 cysteines. So we did the mutation. I think we did this work on Perth and Texas, and we changed these cysteines to either serine or alanine. And then we did the expression, purification. Purification, we used our established process, and Kathy and the team, they were able to purify the protein quite nicely. Here is the data I want to show you. That first question that we asked, that because we are changing these normal or natural cysteines to serine or alanine, will it impact the expression and the purity and the yield? And I think the answer is that it did not really impact the yield. The proteins were expressed equal or slightly higher levels compared to the wild type. So that was the first test we did. The next, we look at the purity. And again, I think if you look at it, I think here are the 2 cysteines, 5 cysteines, and the 3 cysteines in the left-hand panel compared to the wild type. They are all getting highly purified band. They are about 95 to 98% pure. Then we said, okay, their purity looks good, yield looks good. Do they have the same trypsin resistance profile? And that's the data Kathy and the team has generated, and I'm presenting it into the panel B. And if you look at it, this is the wild type. The first 4 lanes are the wild type. Then it's the 2 cysteines, then it's the 3 cysteines, and 5 cysteines. That's what, no, 5 cysteine is first, 3 cysteine. And the first lane is the DS or the protein all by itself. Second, we added the trypsin. Third, we added heat. And the fourth, we added the heat and trypsin. And I think you will see that the protein degrades. So I think they were comparable to the wild type. And so by changing the cysteines, we did not really impact the trypsin resistance profile as well. So this is the remarkable data that we have observed. I think what we have done, I think we took the protein and then analyzed it on the size exclusion. As you will see, the retention time for all these proteins, either wild type or the 2, 3, and 5 cysteines, they are all comparable. When we have taken them and started seeing it, what happens is if you treat this protein with the detergent, and that's where I'm showing you. So I think this is what the rosette structure looks like. Then when we treat with the detergent, the rosettes are dissociated into the trimers and the peak shift. to the right, means smaller particles. But there was a small portion that remained in that confirmation that we were, no matter how much estrogen treatment we gave, we were not able to move it to the trimers, and that's what probably the reason initially that we thought that that's the reason why we are losing the potency. On the other hand, if we take the 5-cis protein, this is what the protein looks all by itself, and then we did the estrogen treatment, you see that it's a almost 100% switch to the trimer. So that's the first evidence that the cysteines are really preventing the aggregation, formation, or multiple formation. So then I think Kathy and the team, what they did, they took all these 4 proteins and then did a differential scanning fluorimetry to determine the TM. And the TM for all 4 proteins are in the similar range. I think it's 55 degree to 56 degrees. I think so. That's telling us the secondary structure is intact and the proteins are in the correct conformation. So here, I think we wanted to look at the DLS, the particle size, and the team has done 3 measurements. We did at day 0 after purification, then we took 1 week, and then we did day 28. As I mentioned it to you before, I would like to capture it here again because we saw the initial loss in H3 potency. That's the reason why we were doing it up to day 28 to see that what phenomena we are seeing it. Is it more larger particles are coming or not? But again, I think what we are showing you here is that compared to wild type and the 5-cis, they are comparable in terms of the particle size, in terms of this is the 38 nanometer particle we are getting it. So at this time and point, we just started asking the question, what will happen? I think happening in terms of the multimer formation. So we hear the proteins are analyzed in the non-reducing and reducing conditions. So this is what is non-reducing. That's where you will start seeing the ladder. The protein will form the multimers, and these are form the ladders. In the reducing condition, it comes out intact here. The first line is wild type. The second is 2 cysteines. And similarly here, it's the 5 cysteines and 3 cysteines in non-reducing and reducing condition. You will see that the wild type and 2 cysteines variant, it is behaving somewhat similarly, and we are seeing a number of conformations or the ladder formation. This is the intact main protein, but these are also being formed. In case of 5 cysteines and 3 cysteine, you are not seeing any of the multiple formation. So that the phenomena that we saw before, I think it is being taken care of it, that that cysteine that was getting oxidized and causing the multiple formation will not be able to do so once we introduce the variant. On the this panel, what we are showing you here is we just took the profile that I think in order to see that how many smaller forms of the proteins or the larger forms of the protein are present, and that's the data is captured here. In the B panel, what we did, we took the ratio between the HA main band, that we call it HA0, and then we also look at it that how many other forms are, what is the ratio, and that's what the data is presented here in the right hand side panel B. You will see that the 5 cysteines and 3 cysteines, we are not seeing anything. It's a bottom, no multimer formation. But if you look at it, 2 cysteines and the wild type, you are seeing a lot of multimer formation. So that's again, by replacing the cysteines, we are able to make a much more homogeneous material. Next, the team, I think Kathy and the team, what they did, they wanted to look at it if there is any other heterogeneity that we can detect. So we started doing the reverse phase column, and in the reverse phase, if you take the wild type, we are getting these 3 type of structures or we are getting it. Similar structures we are observed when we did it for two cysteines. But if you look at it, the three cysteines or five cysteines, we are getting one homogeneous peak. And again, I think this study was done at 2 days, two different time points. One is the day 0, another is bottom panel is day 28. Even keeping it for 28 days at that temperature in those conditions, we are not seeing any type of structural changes. We are getting the homogeneous material. So then what we also wanted to look at it at this time and point, we are satisfied that by making the cysteine variants, we are not compromising the structure. The structure is stable, and then we put it on the stability. In the stability, we did it at the 5 degree and also at 25 degrees. What you will see here, I think in the upper panel, in the red one, this is what the five cysteines, and this is the wild type. You will see that by um, about a year, I think you see about 60% loss in potency if we take wild type one, and in this in the 516, the potency is not really driving that fast, even at the 4 degree of 5 degrees. So it's very stable. There are little bit fluctuations here, and that is probably can account for the assay variability. Then we put the protein again at 25 degrees. Our goal was to see if it is going to be stable at 25 degrees. And here you will see the dramatic effect. This is the wild type. I think within about 2 weeks, you were seeing a huge loss in potency. It goes from about 40 to 40%. And then if you leave it longer, I think it keeps coming down, and then we lost about 80% of the potency of the molecule. While if we take the five cysteines, I think the protein remains rock solid at 25 degrees for 400 days. And that was the limit of our time point. I think we could have gone at that time, could have gone beyond, but we stopped here around 300, 365, 380 days. So here on the B panel, what we are showing you here, that the drop in potency, what we are seeing in 25 degrees, it is correlated with the formation of this is the 5 degree data. This is 25-degree data. I think in 25 degrees, you are going to be seeing more. And at 5 degrees, you will see less multimer formation, but this directly correlates with the loss in potency. So this is what we have shown, that I think with this approach, we were able to, by mutating the cysteines, we were able to make a vaccine, or at least H3, which was temperature thermostable. Next, I think slide. Yeah, so I think we did more analysis. We took the same samples that were stored at 5 degrees and 25 degrees, and this is the wild type. This is the um, 5 cis, and we compared day 0, and I think it's 12 months stability, and you will see that there is no change in the profile of the protein. 5-cysteine variant remains stable for 12 months. The same is, this is 5 degrees, this is 25 degree. It's much more dramatic. I think in the 25 degrees, what you are seeing it in 12 months, I think all those things, the smaller peaks are almost to the base level, while the 5-cis variant protein is remained strong. So this is the data that we have generated so far. Again, I think showing you in comparison the stability profile again in a different format. The top line is the stability for the 5-cis, and then goes up to 2 cysteines, and then this is the wild type is totally gone, while the 5-cis variant is um, literally no loss in the potency, and this is all done by SRI-DSA. So next, I think what the team has done, They did an immunization. They immunized the mice with the wild type and 5 cis variant in the mice. In that one, I think you will see that I think B and A, B's are usually the 5 cis and A's are the wild type. This is the first time point, this is the second time point, two time point. But you will see that if you look at the sera that were tested for the HAI activity, HAI activity is not compromised by the cis variant. So that's again that we are not inducing, we are not really impacting the induction of the functional antibody response. So protein is stable and we are inducing functional antibody response. So that's what the approach I want to share with you. These are the conclusions. I think probably the most significant part is by changing the cysteine, we were able to create a molecule that was stable at room temperature for about a year. And I can go to the next part of the presentation. If you have any question you want to ask now, or we can go to later.
I think, Indresh, it would be good to continue your
Okay,
presentation and then take all the questions at the end.
thank you, Manon. Thank you. So this part, I think we just want to go back, go and show you some examples for the lyophilization and formulation development. I think by any means this last list is not complete. I just wanted to share a flavor that the things are being worked out in different contexts for different combinations, and it looks quite encouraging. I think the goal is going to be that can we develop a liquid vaccine just by tweaking the formulation. And the other goal is going to be, can we just take those formulations and lyophilize it? And I think I will show you a couple of examples here in the sections. So I will show you the lyophilize and formulation. I think the lyophilization, I think we all um, know that I think it is very good in terms of preserving the activity of the protein, provided it's done right in terms of the condition. It is a simple process, but yet is quite complicated. There are several steps, several stages for the lyophilization, and I think without boring you with too much detail, I just want to show you the basic process. It has a freezing step, and then it has the two drawing steps. So that's what I think I identified here. In terms of the first drawing step, what we want to do is to remove the unbound water molecules from the protein. In the second one, in the second drying step, what we do is to remove the water molecules that are bound to the protein. So these are the two very critical, and I think there's a lot of optimization that needs to be done in order to get a cake that is going to be stable, that is going to be the water content is going to be fairly low, and on top of that, it also induces the quality of responses that we are looking for it. So going to the next, I think I will show you. So this is the data that was published by Flood et al. And what he has done, he has taken that H1N1 influenza subunit vaccine and tried with a couple of formulations. I think he has done disaccharides such as sucrose and trehalose. He used different combination, different concentration, also included glycine in addition to surfactant and phosphate buffer. So that's what I think he looked at it, that formulation. But he also did the lyophilization of the protein in that formulation. And what you will see here, I think it is quite amazing that we are not losing any activity. The protein is fairly very stable at 30 degrees, 37 degrees, and 48 degrees. These are the two temperatures that the team has looked at it, and there is very little change, if any. I think there is going to be some preferential factors if you use sucrose or trehalose, But other than that, I think it is just very, very encouraging to see that the vaccine was stable. I think in the article, I think he was saying it at about 40 months, the vaccine was stable. So that's one example which is shown here. And I think at my time at the protein sciences days, I think the team also worked on H5N1, and we showed the lyophilization was quite good in stabilizing the protein. We had the cake. We are looking at the moisture content. We are looking at the the TMs of the cake, looking at the color, looking at the collapsibility. We did a lot of characterization. Again, that work was supported under the BARDA contract that Protein Sciences had it. So it is good confirmation of that one. This is another remarkable study that I found. I think this is by To et al. And what they were doing, they showed the work on the filovirus. This is with the Ebola and Marburg virus. They developed the thermostable formulation. They lyophilized those two formulations and tested them into the cynomolgus monkeys. So they established that yes, the formulation is intact, and then they were able to immunize the monkeys. And post immunization, they challenged them. What you will see here is that all the monkeys that were vaccinated, they survived. Only the controls they were able to um, they did not survive. Then we are looking at the clinical score. You will see the clinical score only in controls, no clinical sign and symptom in terms of vaccinated animals. Here we are looking at it. Um, so this is what the viremia is, and you will see the viremia essentially in control, and no in the vaccinated animals, and also the loss of body weight. So loss of weight was shown in the controls, and then not in the vaccinated ones. So again, I think it's a very good um, study, and that again proves the point that yes, it is possible to make the thermostable vaccine. Of course, I think optimization of the lyophilization conditions and the formulation will take time and effort. The other example that I want to show you, this is the work that was published by Kramer et al. from the Infectious Disease Institute, and I think they were working on the TB vaccine. In the TB vaccine, Erin, I think what Ryan has done, that he has tested again different formulations, and the formulations were again contains disaccharide or a combination of disaccharides and mannitol that can achieve substantially improved thermostability. And I think that's what the data here is. He's also mixed it with the GLA. So here he is looking for it that once you make a vaccine with the GLA, what the loss of GLA function and activity is. And again, I think it looks quite impressive that we are able to get it a thermostable, room temperature thermostable formulation. So this is for the TB vaccine. Another example, I have it for the rabies vaccine. In the rabies vaccine, the work that Dr. Randolph and colleagues are doing at University of Colorado, I think is quite amazing. What they have done, they have taken an inactivated rabies vaccine in a spray dried spray dried from the polysaccharide solution to create a glassy microparticle. Then they added silica on top of that, and then they look at the stability. I think they were able to show that the vaccine is stable for 3 months as at temperatures as high as 50 degrees, which is remarkable. They are also going to be doing some more studies, and I think I was looking at their press release in terms of what the path is going to be. They need to just go and do some animal studies, and after that, they will probably go and do phase one, but the particles looks beautiful. Another advantage for these particles are that this is a control release particle, so you can actually control the release of the rabies vaccine by manipulating the silica content. I think so. That's what is very, very appealing to me as well. So next, I just wanted to show that another approach that the investigators are using in the field is patch technology, the needleless delivery. It has several advantages, but more importantly, I think the way these needles are formed, you are exposing the proteins to a higher temperature, and then I think the teams have done the stability. So I think I just put the number of type of needles are used in the field: the solid microneedles, hollow microneedles, coated microneedle, dissolving microneedles, swelling microneedles, and then porous. There are a lot of literature is available here, but I think in the study that was referred to, there is a 5-in-1 microneedle patch incorporating diphtheria, tetanus, pertussis, hepatitis B, and influenza type B vaccine. I think that vaccine was shown to be stable for 12 months at 25 degrees. So that's what they study. Other study that I am showing you here is this is our dissolving microneedles. This is our for the flu program. I think it's the two A's and the B strains that were used, and you will see that I think they are stable for about 6 months. I think in different forms. So that's another. It's a dissolving microneedle, and probably yeah. So this is. The last part is again, I think this is a Vaxis study. I think they have done high density microarray technology, and I think using that technology, they are showing that the vaccine is stable for up to probably 12 months at 40 degrees, and that's what they are starting the clinical trial. I think probably um, may know a little bit more, but I think that's what also being published. So there are a number of technologies are available, and now it's time to focus and see what we can use. I think everyone, all of us, I think in industry and academia will have our favorite method to pursue. So having captured this, and then I think what I have shown you all the things that I said I will show you, but in my mind, I think the summary is, as I said, that the technologies are available both in academia and industry, and now I think it just need to probably um, decide and focus on developing the stable thermostable vaccines. I think that one of the projections that I was looking at it, I think the thermostable vaccine market is going to be expanding quite a bit in the next 10 years. And I think that suggests that the number of teams and organizations are already working on it. So I can stop here. Thank you very much for listening. And thank you very much to Manon for giving an opportunity.
Well, Indresh, thank you very much for giving this overview. It did bring back a lot of old memories because at
Yes.
Protein Sciences we were obviously struggling with the fact that the hemagglutinin, especially the H3 versions of hemagglutinin, were highly prone to aggregation, and that really impacted the development of that influenza vaccine. Now,
Yep.
you showed a tremendous body of physical, biochemical, data and how we looked at things. And maybe you can tell the audience a little bit, as we're waiting for other questions to arrive, tell the audience a little bit how we obviously went to the FDA with all this data, and the FDA is very open-minded in using these biochemical characterization methods to accept, for example, going from one scale of manufacturing to a larger scale of manufacturing. But maybe you can comment a little bit on how they viewed the changes that we made and whether we were allowed to use them, yes or no.
Thank you, Manu. I mean, probably I will comment, but I will definitely will look up to you to probably complete the story because I think we were together in that thing. I think FDA, I think we were very receptive, very supportive. And also BARDA was very receptive and supportive of the data. And then moving into probably implementing it in the commercial space. I think if I remember correctly, I think FDA was very supportive, but they wanted us to do a clinical bridging study.
Yeah, and I maybe a comment from me. It
Yep,
was quite disappointing indeed that they did not allow us to use these mutants because
yep.
I do think coming at genetic engineering as a solution for dealing with thermostability would be a nice and elegant solution, but they felt that we were possibly basically optimizing the molecule so that it would perform better in the potency assays. And they did not like that. And it was very surprising to me. And I'm just wondering whether that opinion nowadays would have changed. But obviously, I don't know that. And I was wondering whether you, maybe based on your more recent interaction with the agency, might have some views on that.
Yeah, I think so. Probably, I think with our interaction with the FDA, we did really provided. This is the body of data we shared with them that we are not really optimizing the assay. We are optimizing the molecule to perform the way we are expecting it. I think probably also in the recent interactions with FDA, I think, and I'm also um, understanding that they, at least not this last year, but before that, I think they are going to be looking at it more objectively, if I may say. Um,
Right.
Again, I think the proof is going to be in the pudding. I think we need to put forward a very strong data package and then provide the guidance in terms of whatever questions or challenges that they will send it to us or put in front of us. But
Right.
again, I mean, this is the remarkable work that the team has done when we were at the Protein Sciences, of course, under your guidance and support. But it was amazing work. We just took it from scratch, and then we just completed the whole story.
Right. We have a couple of questions from the audience, and
Yep.
maybe if that can open the microphone for David Whitaker so that he has an opportunity to ask his question. David, I think you're on mute. I can't, David, you are still on mute, so we can't hear your question. I cannot unmute you. You need to do that yourself. And otherwise, at the same time, can we already open the microphone for Ab Osterhaus? He also has a question.
Yeah, I hope you can hear me.
Yeah, we can.
I can
Thank
hear you.
you.
Yeah, thank you.
Well, Indresh, again, as usual, a fantastic story, of course. Very impressive data on flu. And I've always been kind of jealous there because whatever we tried with, for instance, polio vaccine development, it was always impossible to freeze dry that,
Mm.
let alone the messenger RNA vaccine. So you were telling us all the success stories about flu, but can you tell us what is the critical point there? Why can't we use, for instance, drying as a universal technology?
So the simple answer is that the drying. I think the technology at a small scale is very attractive, beautiful. But scaling up the technology, I think it has little bit of some challenges, and I think we need to make some technical advancement in terms of scale up. I think when I was at Protein Sciences, I think David Rhodes had made some connections, and I think we were able to move from vial to the tray. And in order to commercialize the vaccine, I think vials may not be an option. But then, what could be the option? Can we just do the large freeze-dried cake, and then we take it and then reconstitute in the liquid. These are the dilemmas we need to work on, or the questions we need to work on it. So that's the reason why I think the teams are also looking at simple liquid vaccine using the formulation trick. So and that's what is happening in the field. People are coming up. I think the teams are coming up with some very potent formulations. So,
Okay, so up, it looks like Indresh doesn't have the question to your or the real answer to your question, but I recall from the work that we did on influenza that it was quite complicated to get a good cake that didn't collapse, etc. My goodness, so I think there's so many variables, right, Indresh, that it's very hard to just come up with some kind of universal process.
Yeah,
But
I think if we
it's primarily scaling up. But as you perhaps know, there were huge projects also funded by WHO to make freeze-dried polio vaccines, and you say it's in the scaling up, but even at the small scale, similar to foot and mouth disease virus, it didn't work. So, and these are extremely stable viruses, So the question is, why? I'm not a chemist, so I'm really I love your story, because it's all successes, but I've been confronted with failures as well. So
yeah,
you are correct. You are absolutely correct. And I think, as I said in the beginning, I think how you freeze and how you set up your drying parameters, that has a huge impact. So I think, I mean, again, I did it in my past life. I think we have done a lot of lyophilization, and again, I think I would love to have a universal procedure, but again, I think it's going to be so much dependent on the target, what target protein you are going to be using it. And I think to a certain extent, if we come up with a better formulation, I think it may address it. But again, may is a big word.
and maybe that micropatch technology that you discussed might offer solutions there that may be more universal.
More universal, yeah.
Yeah, thank you very much, Ab. Maybe we can move on to some of the other questions that we have in the chat. Indresh, can you discuss how the stability, or rather lack thereof, compares between membrane-bound hemagglutinin versus soluble hemagglutinin?
I think I would say my experience is with the membrane bound, because I think that's a backup platform that we use at the Protein Sciences. It is probably, if I may not be overestimating, but I think this is the only system that can give you a full length protein can be expressed. And when I was working on HIV, I struggled to express the intact envelope protein, and I was not able to. So I think I have more experience with this membrane-bound protein in the correct conformation. In terms of the stability of the liquid form or the secreted form of the protein, I think somehow you need to find a way to hold the protein together. And that's where the transmembrane domain plays a role. And I think if you look at it, I think the cysteines that we shared the information with you, we are seeing the cysteines, two cysteines in the transmembrane, three in the cytoplasmic tail. We did not really get to dissect exactly how the folding takes place. But my feeling is that if you go with the secreted form of the protein, the structure is going to be a little bit more, how to say, unstructured is not the right word, but I think it's not going to be very tight or intact. So it may impact the stability.
Indresh, maybe if I may chime in, we did in the early days at Protein Sciences produce protein hemagglutinin without the transmembrane domain, and it did not result in functional antibodies. So it was very stable, and it really aligns very nicely with what you described for the cysteines and how removal of them resulted in better stability. So what we saw is it was more stable, was much more stable, but it did not result in functional antibodies, which is why that approach was abandoned early on.
Yeah, Yeah.
So if I move on to the next question from the audience, is it necessary to develop the thermostable vaccine at room temperature if 4 degrees works? And I guess, you can comment on that.
Yeah, so my view is that I think 4 degrees, I think you're going to be constrained in terms of I'm not looking at the US and Europe, but if you go to the developing countries, I think that's where the infrastructure is not going to be supporting the 4-degree vaccine, because I think this cold chain is going to be a constraint in vaccine distribution. You can get to a central place and store the vaccine at the 4 degrees, but when you go to the field, it will not be able to support it. I can tell you by my experience in India that I mean, we were not able to at the center. Yes, it is stored at 4 degrees, but when you go to the field for the vaccine clinics and all those things, it is not 4 degrees at all. So that is the reason why having 25 room temperature is stable, or even 30 degree or 37 degree stable vaccine will probably give us a huge advantage.
Indresh, as a next question, do you know of any examples where the introduction of cysteine residues can be used to stabilize protein vaccines? So not removal, but introduction.
I don't know, any example where cysteines are introduced. I think I'm always looking at it to remove the cysteines. I think my challenge has always been that to determine if there is an unpaired cysteine, that is what is going to be causing most of the challenges.
Right.
So I'm not aware. I think if all the cysteines are properly or correctly paired, then I think it's will result probably not that type of situation. But generally speaking, I think you will find one or two culprits somewhere.
Okay, so the next question really relates to your current employer as well. Thank you very much for your talk. A question from Wendy Crocker. Would you have any views on improving the thermostability of adjuvants such as Matrix M to formulate an adjuvanted thermostable vaccine?
Yeah, so I think I will not probably comment the strategy that Novavax is using, but in general, I think I can tell you from my previous experiences that yes, adjuvants can be lyophilized, they can be reconstituted, and they will maintain their adjuvant properties. And hopefully, Manon has invited our head of R&D to speak in ISV meeting on the adjuvants. I think she may touch upon it.
Okay, so you're actually saying that at our conference in Antwerp later this year, Roxanne will be able to address that question in greater detail in person?
She may touch. I have not really talked to her what she will be presenting, but she will be. She is very interested in presenting the adjuvant approach on adjuvants. Yeah, so she may
Okay,
touch upon.
Wendy. So I invite you to attend that meeting, and then you'll get your question answered from an
Answered by
anonymous.
the boss.
Yeah, from an anonymous attendee. Brilliant talk. The effects of the increased temperature to vaccine stability and efficacy is true. The lyophilization process helps to protect the vaccine. It has been tested, and then he gives you an example in Science Direct. It's in the chat. I'm not familiar of the article. Maybe you are, Indresh, and otherwise it might be something good to look into. I don't think it's really a question, but more of an example for you to take a look at another
Absolutely.
piece of literature. David McElhale asks, concerning the influenza vaccine potency assay, could you please elaborate on the potency assay itself to help put the FDA's concern into more context?
I don't know, Manon. Maybe you take that.
Yeah, we're talking here, I guess, about the SHRIT assay.
SHRIT assay, yeah.
Could you elaborate on the potency assay itself to help put the FDA's concern into more context?
I really don't know. I think probably you. I think SRID assay is very well known. It's a simple phenomenon. And
Yeah, so
also
it's,
yeah, go
yeah.
ahead.
Yeah, it's basically an assay that is that simulates the performance of the antibodies, right, in vitro. And
Yep.
but I don't know how we can address, how that would put the FDA's context into FDA's concern into more context. I
Yeah.
think. Yeah, they were just concerned that making these changes may have had, and maybe the aggregation of the H3 molecule actually contributed to the really good performance of flu block in vivo in humans. And they were actually probably concerned that you're now showing that your product is much more stable, but maybe because you don't form these larger aggregated particles, maybe that will impact the actual performance of the vaccine in humans. I guess that's all I can say to
Yeah.
that question, Indresh. I don't know.
No, you're right. I think, you know these things a little bit better than me. So I think, yeah, absolutely, your comment makes sense. But I think in terms of optimization of the assay, I don't know. We haven't really changed the assay. When we were doing the assay, we are running them in parallel, and we were looking at the performance. And I think it's a known phenomena that the diffusion will not take place at the same way if you have a more aggregated or multimeric protein compared to a uniform, stable molecule. That's what I think.
I think as a last question from the audience, can some AI tool help solve some of the problems associated with the different steps of lyophilization? Artificial intelligence, can it help
Yeah,
solve some of the problems that we encountered?
sure. I think yes, it can help. Also, but in order for AI to help us, we need to have large body of data. Then they can do the analysis and recommend based on the structure and the thermal stability. I think so. We are still at the first stage. We are generating the data before it can go to the AI. AI will not work without having some data. So we need to provide large body of data, and then it's going to be predictions that can be utilized using AI.
Okay. All right. Well, I guess that we have addressed all the questions from the audience at this moment in time. Oh, wait a
Thank
minute.
you.
There's one more, Indresh, one more. What laboratory methods are used to evaluate thermal stability and potency of thermal stable vaccines? Question from Tahir Shah.
Manon, what is the question?
What laboratory methods are used to evaluate the thermal stability and potency of thermostable vaccines? I guess we go,
Yes.
yeah.
Yeah. So essentially, I think I will do the same things what I have shown, that what we have did, what Kathy has done. We are going to be looking at the structure. We are going to be looking at the conformation. If there are known epitopes, if there are monoclonal antibodies are available for the known epitopes, then I think I will use them. If we have the neutralizing antibodies, then we can test them. There are a whole lot of things you can do, but I think I will probably focus more on biophysical, biochemical first, then go into the immunological aspects. I think if the structure is correct, is appropriate, then we can look at the functionality.
Okay, well, Indresh, I would like to thank you very much for your overview and for sharing this important data with us. For those of you who have a few more minutes, I'd like to run through some of the activities that ISB is undertaking, and really encourage you to participate in the future. So we will have our annual congress this year in Antwerp from the 12th to the 14th of October. The chairs, Michael Schopsaerts, who is chair, with his co-chairs Lakshmi Krishnan and Isabella Rules, are putting together a wonderful program. We already have gone live on our website with an overview of speakers that are confirmed, and we highly encourage you to sign up. It promises to be a nice meeting. Next slide, please. So we are also able to provide some travel awards to early stage researchers, meaning students and postdocs with up to 5-year experience for students both from LMIC countries, but also for students from other countries. So take a look at the website. The deadline, the application deadline is fairly soon. It's March 31st. So I really encourage you to go take a look and see whether you are eligible and can apply for such an award. Next slide. All the information can be found on the website. We also have a call for abstracts open. The deadline is June 12 to 26. So half of our talks are being selected by having invited speakers, and half of the talks will be selected from abstracts that are being submitted. Next slide, please. And there, you can find again on the website all the information on what the various sessions are about and where you could best apply. We have in April another webinar by our 2025 fellow, Dr. Baiklin Seong from Yonsei University in South Korea. And he has agreed to get up really early so that the seminar can actually take place at a time where we are awake. So that's really wonderful. The seminar will be moderated by June Ray, and he will be discussing chaperones for artificial intelligence, which is obviously a hot topic, and structure-based vaccine design. So this will take place on April 8th at 4 p.m. EDT. We are having our next ISV Industry Mini Symposium, which has been organized by Corey Fang from AstraZeneca. He basically put a topic together around AI use in vaccine development. We're going to have speakers from Novavax, Kumar Singh, from Evaxion, from AstraZeneca, and Epivax, all companies that are very heavily into this field. So this will take place on April 23rd from 9 to 11. Next slide, please. We also have a whole series around next generation vaccinologists, where young scientists will share their research, and the next one is going to take place on April 29th. And we'll have 2 speakers there, Alicidia Ramos Barros from Switzerland and Kanita Patakul from Thailand. Next slide, please. So I want to thank all the sponsors and partners of our organization, which makes it possible to provide these awards, to arrange all these conferences and seminars and webinars. So thank you very much to all our sponsors. Next slide. We have a lot of fancy QR codes where you can find news on the society on various social media channels. And there we will update you also on latest news, but also our website is really updated on a very regular basis. So please feel free to log on to that. And if you aren't a member of ISV yet, we encourage you to join ISV. There are a lot of benefits. I want to today specifically point out the mentorship program. You get discounts to attend the annual meeting, but the mentorship program is specifically for young scientists who are then being connected with more experienced scientists. And this program is really off to a great start. And in our upcoming newsletter, you can read more about that by Linda Lua. Next slide. Those were the slides. So thank you very much for your attention. Thank you so much, Indresh, for your talk and for providing, a great overview on all these complicated methods that we can use to actually evaluate vaccines. So looking forward to seeing all of you at our next seminar. And again, thank you for your participation today.
Thank you, Manon. Thanks, everyone.